So, well,
I
real
know
quick,
it's a little dark, but
we have a chrysogorgid, and there's a squat lobster in there.
It's a crisis.
So a small little arthropod.
Yeah.
Oh,
It's made its home.
hello.
Oh,
Oh, I
Yep.
love these corals.
oh,
so pretty.
How's that, Greg?
If only it was
Looks good.
purple.
It's beautiful.
Can we do a full zoom?
sure.
So the squat
Going
lobster
for
that's
it.
living in the coral, this isn't, oh wow, we're right on up in there.
Yeah.
That's,
Look at this. So this is a
Howard
good
zoom.
chance to look at the octocoral polyps
Yeah.
too.
Oh my god.
So you can really see the 8 tentacles around
Yeah.
each one. And they have
Oh.
those pinnate, they're pinnate, so they have those little projections coming off
Yes.
each tentacle.
Is there like a lucky tentacle with like 9 or 7?
Oh my gosh.
Does
A
it
7-leaf
exist?
clover.
Yeah.
we're
There's
gonna be
7
here a while,
tentacles.
guys.
All right, video, let's come full wide. It's time to go.
Coming full
So
wide.
the lobster, unlike, you know, the fishies that are
Oops,
stuck.
sorry.
Oh, wow.
The
No
shrimp.
worries.
Or sorry, the shrimp that are stuck inside. This guy, he can leave. that
Yeah,
coral
yeah.
probably, yeah.
Or
But-
she.
Yes,
Right. Unlike
Or
the
there.
shrimp in the sponge.
yes.
Stuck forever. We should go back and free them.
Just go on a rogue mission, free all the
We're
shrimp.
full wide, Greg.
There's a
Thank you.
nice coral.
Wait, so did you answer the...
I didn't, but the
That's
coral
awesome.
coral tree graphing question. So we can't like graph the skeletons together, but you do often see in really areas with dense corals that corals will grow on top of each other. So they'll kind of just start growing wherever they can. So if there's a piece of bare skeleton, a new coral might settle on that and just start growing off of it. So it kind of looks like they're
Yeah.
growing together. Or we have seen a couple times in areas where there's hard corals growing in the Atlantic, you have like 2 different species of hard coral that are starting to fuse together. And so we're not quite sure if they're sharing resources or not, but it's interesting to think about because they're different species, but are they able to essentially coexist without without there being competition there? Because when you look at some corals, if you see like 2 corals growing into each other, you'll see that they will essentially evade each other because all corals are cnidarians, which have stinging cells.
Oh,
So they can sting each other
wow.
if they're, or anything else.
But if they're the same species and they're kind of growing towards each other, they'll just sort of grow around each other.
yeah.
And mingle.
Really?
Yeah.
Alexis, is the stinging mechanism, that's how they get their food down here, right?
Yeah, so they're called cnidocytes. They're like these little, they're these little cells, and there's essentially like a little arrow on like a little barb on a string. And so they release that and it stings something, and it can either like stun it or paralyze it. These octocorals don't have, to us, they're not really strong stinging cells, but to something really small, obviously, it would be more impactful. Like when we handle corals, we don't get stung by them, but other organisms would be more sensitive to it.
And what types of organisms would that be?
Like small plankton, that's
Okay.
in or like even potentially some of the animals that land on them that are associates. But it just, yeah.
Which everything you just said sounds a lot like trees. Because, you know, like people...
I've never met a tree with a barb on a rope.
Right?
Yeah,
Never been
trees have
harpooned by a
stinging
tree.
cells. No.
That would be cool if trees had stinging cells, though.
But the idea of grafting, I mean, trees do the same kind of thing. Like
Yeah.
they'll like grow into each other, not, you know, people can make them grow and graft them, but
Right.
they'll just look for the
I
light.
think for deep water corals, we still don't know enough about them because we're still having difficulties keeping deep water corals alive in labs, if I remember correctly.
Yeah, that's definitely true. There's a couple species that are pretty hardy that we can keep alive, but most of them, even if we try and keep the conditions the same, they just don't do super well and they die after a couple months at max. We think some of it has to do with current flow, so like they are really sensitive to, they need high flow. So there's been some people that have had more success when they build like cylindrical tanks instead of a tank that's like rectangular, so it has more consistent flow all the way around. But
how big are tanks like this?
I mean, we have ones in our lab for like Lophelia, which aren't. I mean, they're 100 gallons. They're not like giant. But aquariums do it sometimes. Some aquariums have deep sea corals.
Do people study like really local what the currents are like and how they change for different coral sites, like one on a ledge versus one that's like maybe more tucked in, or is it more of a larger, like broader current study for the region?
Yeah, I think for the most part it's larger current studies for the region, but it's not to say that it wouldn't be like really interesting to put some like flow meters down and like get actual readings. And I'm sure some people have somewhere. I just am not privy to it, but it's, since it's so important to their overall health, it's definitely something that we should be considering for long-term studies.
Yeah, I remember being surprised at how elusive it is to kind of get the finer scale current motion down here, that data.
Yeah.
Because even on Herc, it's not like you can really subtract out your motion easily.
Right. No.
When it's ripping, you get a sense.
I heard subtract out your emotions. And
sometimes you got to do that too.
Yeah. Herc has a hard time with that.
What about things like pressure? How do they recreate that?
So actually, it turns out that most of these corals, they are not sensitive to the pressure change, at least not in a way that we're able to observe. They don't show signs of stress when they come to the surface due to pressure. Yeah, so that there is a certain depth range where, so past 1,000 meters, they're harder to keep alive, and we don't know if that's pressure related or other things. But when you bring them up, it's not like fish obviously have swim bladders, so they can't come up without being disrupted. But corals don't have any like air pockets in them, so there's not anything to alter their morphology as they come to the surface.
They don't have to decompress.
Right.
I envy them.
I know, me too.
Can you imagine how long you'd have to decompress for if you were a human at this depth?
Oh, God.
All right, Marley, have fun.
Yay, here we go, here we go.
Ooh, that's a nice view, right into that spiral.
We're going in, y'all. And I'm gonna get it in focus.
Oh, this is mesmerizing.
Yeah. You want to turn the lasers off for a quick beauty
shot.
Wow.
Ooh.
Oh. Just exploring.
Wow.
I love how much color we're able to get off the Herc cameras. Because these cameras, they have like a separate sensor for each of the...
Yep.
Yeah.
Yeah, I'm starting to lose my grounding, so let's come slow wide.
Coming slow wide. Kind of slow.
Do you want me to hold off on the ship?
No, we're gonna...
All right, we're full wide.
Thank you. It's time to bounce.
Oh, it's something
nice
real tall up there.
It's like some bamboo corals. Those sponges that are kind of like rod shaped like that, those are wisteria.
A viewer's wondering if we could talk through how the laser pointer system works and how we gauge distance.
Oh.
That's an excellent question. Our two lasers are 10 centimeters apart, and they are mounted on top of our main camera, and they're parallel to the camera. I'm being fed this information for the first time. Can you tell?
You're crushing it, girl.
Right. So obviously anything that crosses in front of the lasers is 10 centimeters apart. It's very helpful for figuring out how large things are down here because we have no real sense of scale. And everything looks bigger underwater.
And in the river mirrors of the ROVs.
Yep. Objects in mirrors are closer than they appear.
I like sometimes when we're sitting on one kind of dense biogenic spot and we're like super zoomed in, and then all of a sudden we zoom out. I was like, oh, that was small.
Yeah, yeah.
You kind of lose sense of that sometimes.
Especially on the huge monitors.
Yeah.
Yeah.
Life's a fractal.
Getting deep back there, yeah.
Always.
1,700 meters deep. Oh, no. It's too early.
It's the return of the
Never
puns.
too early.
Oh, there's a cool
Deep
little
sea
sponge.
philosophy.
That's a little sponge right there.
Oh.
That's a weird
Yeah, it's
shape to that
interesting.
sponge.
Hi.
Video that's pushing.
It's like a sad little umbrella that isn't quite working right.
Hold that.
That's a
Droopy
giant
umbrella.
mushroom.
Droopy umbrella.
Okay, let's push in more.
It's very
Hey,
long.
hip,
Pushing in.
What's the name of this species?
I do not know, but
Unnamed. Just like
That's
this seamount.
full.
it's such a small little bulb at the top. That's
Yeah.
what's surprising to me. Normally they're a little bit bigger.
I know, it's funny. You invest so much material in the stock and so little
Right.
in the capture mechanism.
Right.
Guess
Okay,
that
let's
works.
come wide video.
Coming wide.
Yeah, so
That's
these
full wide.
sponges like that are
Massive
filtering
sea
nutrients
star.
out from the water through that top cap, right?
Yeah, yeah. So they pull water through.
Oh.
Do we want a massive sea star?
I don't think we want a massive. This
Oh.
is a slime star, so literally
Slime.
it would be like a bucket of goop if we brought that to the surface.
Yeah, we don't
We've
need
I'm
done
that.
into it.
We've done hagfish before.
Dennis
I mean,
into
Oh, have
it.
you
The
really?
data team's like,
Like, we don't need that.
No thanks.
Did Steve just say
Right.
that this Calophacus, is that the sponge we
I
just
think
saw?
so, yeah.
Okay, cool.
Calophacus.
Calophacus. I like it.
I think this is that bump.
Yeah, exactly. It's real.
Trust but verify.
Yeah, so this, it's interesting. This whole outcrop is like 127 meters across of the same contour, which is quite wider than everything before it.
Yeah, it's a little deceiving. Like, there was a ledge that we had earlier that was so flat with sediment ripples, whereas this one, it's so 3D, it's kind of hard to tell that we're
Right.
on such a
Right, that we're on a
I was
flat
expecting,
ledge.
like,
Yeah.
like a very notable little mound in the middle that we're kind of just to the northeast of, but wasn't quite obvious.
Yeah, me too. Sensor bias.
Yeah.
So when you get off the ridge, do you feel the current pick up a bit, or?
Yeah, they're actually, it's kind of like right as we crest up onto the top of the flat.
Oh, really?
Is it like being on the summit of a mountain? It's all of a sudden windy.
Yeah. Yeah, I can feel the wind in my hair. Hence the hat.
Gotta tie back the hat.
Greg is her. You
Kirk
become.
is Greg.
We're gonna steal bubble.
We'll see, urchin.
Yeah.
Sure.
So I'm going to bring
So
our
that's
ship move
almost
a little.
1.5 meters.
I'm going to bring our bearing a little north.
Yeah.
Okay.
'Cause we,
Get to waypoint 2.
yeah.
How many waypoints are there?
Let me see.
We have 4 waypoints and an end,
Oh,
Terminate.
but we also have a launch and a start. So is that 7 waypoints? I'm not sure. Numbered, we only have 4 though.
got it.
How do our numbers look?
Oh, they're all stable and steady. All good.
Bridge.
So, Alexis, so like this one, you might argue that the polyps have migrated, or that maybe just the bottom has died off?
I don't think the bottom's died off. I think this is just, for this species, they don't have it all the way down. They don't have polyps all the way down. We don't really know when they're juveniles if they start out with polyps across the whole stalk and then they migrate or not, but
Why do they start spiraling at like that halfway point?
I don't know. Good question.
That's where the hips are.
Yeah.
And how do they know it's the halfway point? They're really just starting to dance.
Yeah, yeah.
I like those more and more every time we talk about them then.
So
Spirit
dance and
coral.
tango.
Sounds pretty nice right now.
At 5 in the morning.
Yeah.
Anytime to
Anytime.
tango dance,
Yeah.
exactly.
Any time of day is tango time in Jess's world.
It looks weird. Video, we're going to push in for identification.
All right, pushing in.
Sponge.
Yeah.
Sponge.
How many? Was this ophiuroids and crinoids on the same?
Yeah.
Wow.
Yeah. Looks like some little crinoids and then a number of little ophiuroids as well. It's a cool sponge. It's like a purple.
Yeah.
light
Yeah, do you think
purple.
that's actual pigmentation or is it unhealthy?
No, I think that's just
All right, video, let's come on.
All right, we're
Really
zooming out.
judgy.
I don't know. I'm just concerned, really.
Hey, Alexis, can you put your microphone a little bit closer to your mouth?
Yes, I can.
So we only seem to find fish in the open flat areas.
Mm-hmm. Yeah.
Yeah, see, I'm so confused by where like
Marley,
in
is
this
that better?
flat
Yes, that is
sediment
better. Thank you.
area now compared looking at our map. It's
We
a
should
little bit-
also keep an
Yeah, I
eye
think
out
we
for
might
a
be a
rock.
little-
we're a little
on
off,
in the
yeah.
translation.
That makes sense.
On that axis we're actually on.
Right, and then we're
Right.
going to start our climb right here.
Yeah.
And there's this kind of like mini summit up ahead.
And so
About
we're
200
looking for
meters.
rocks because we want to be able to better date these seamounts and also
Yeah.
understand sort of the mineral composition on the
Yes.
boom.
So I think the seamount's thought to be around 70 million years old, but confirmation of that would come from getting actual like basalt that is making up the seamount.
Yeah.
But they also, all of what we're really seeing is like a manganese crust, so this oxidized, because it builds up over time. It's a really slow process, but I think it's like, what is it, 1 to 10 millimeters per million
We're back
years.
to even tension.
Yeah.
Wow.
Grad school again.
Yeah.
It is strange though, right, that the
Forever
change in
scholar.
tensions.
What are we
But
looking
those-
for
I
that
don't
would,
get the right
would
correlation to
trigger
the
a,
winch tension
collecting
graph there,
a
though.
geological sample then?
It's
I
really
think
just
that
something
that's
that's
actually
loose.
coming off of the
So
overboard
most
shift.
of these are
So it's not going to have any
sedimented
real
to
correlation
the
to there
pavement.
because it's only going to experience
So you
from
can't
the
actually
vehicle
collect them,
to
but
the
if we
last
see
map.
any that look like they're
Yeah.
detached,
Yeah.
those would be good candidates. And we're trying to do
Yeah, it's
one
down below.
rock collection per shift because we're trying to make sure we have a rock sample available for the summit of the seamount.
Someone online was wondering about temperature fluctuations in various seasons down here.
Oh.
Where do I see temperature on here?
I can look at sensors right now. The temp probe reading
2.6.
0.84. That's not correct.
We don't have a
It's on-
temp probe on.
2.64 on the
Yeah.
CTD? Okay.
Yeah, you can see it on the.
You can see it on the sensors page if you.
At this
Toggle over
depth,
to
though,
it.
do we see
This
fluctuations
is, yeah, that's
over
fine.
summer versus winter?
I don't think so.
I think it's pretty cold down here all the time.
But they're not unaffected by kind of like tidal sloshing, I think. That was surprising to me, actually, to realize that even these undercurrents have these tidal rhythms.
Oh,
So Ben, is there a sense then that there's like a seasonality to the currents themselves? So there's not like a seasonality like we would think of on the surface, but is there another type of seasonality that we get in the deep ocean?
Yeah, I'm not sure about that. I know that, your surface currents are definitely affected by
Right.
temperature gradients like that, but it may affect downstream. I really don't know. Again, I am always surprised at how elusive the kind of bottom flow at these fine scales are to quantify.
Yeah.
You kind of really need to deploy these sensors, tilt loggers,
Yeah.
and recover them later. So it's quite a lot of effort to get your, temporal aspects of that flow.
Yeah. Well, and I know so at some of the later dive sites that we have planned outside of Baker Island and Howland Island National Wildlife Refuges, those seamounts, they have the equatorial current is like pushing up against
Yeah.
those seamounts, and that's one of the things, that upwelling of deeper cold air moving up the seamount is one of the things that makes those spaces so productive
Right.
for marine life.
Right.
But then also closer to the surface, that translates all the way up to the seabird colonies that live up at the top of those.
Yeah, that is definitely cool aspects, that the coupling of the bathymetry to the water column dynamics, where
Yeah.
you do get upwelling induced by these features on the seafloor.
Yeah.
And they can be real hotspots for planktonic aggregations and
Yeah.
It's all
kind of
connected.
eddying. Yeah.
No, it is. I mean, you think of the massive expanse of the ocean, and generally there's nothing there for these currents to hit. There's nothing for life to aggregate on, except for these giant seamounts that are like thrusting up from the seafloor. So we have these lines of ancient volcanic seamounts that are not visible from the surface, except for in a few places where you have remnant atolls and islands, a few little spots that now tend to be large seabird colonies. But below the surface, these ancient volcanic remnants are really aggregating life, and that's what's allowing us to be out here and targeting those for discovery and
Yeah.
exploration.
Yeah, and I wonder if having that induced flow at these seamounts actually facilitates recruitment from the plankton stages, because you're spending more time in the vertical there, and then
Yeah.
you have more opportunity to recruit to that site.
Yeah.
So there's a lot of,
Finding a rock here is going to be hard.
If it's not clear, if you were in freshman bio and you did that food web project, this is the team that said, how close am I to a seamount and what was my flow rate? And
We're the word problem of nightmares.
That was not my freshman bio class.
Yeah, and the talk about temperature earlier, most of these habitats down as deep as we are pretty consistent. So we're at 2.67 Celsius, which is also really cold. And it'll be that pretty consistently at this type of depth for all seasons.
Yeah.
But other parts of the world where you have really strong surface currents, so we do some work
Okay.
off of the off of the U.S. Atlantic coast, and the Gulf Stream there is
Where's
a really
waypoint
strong
3?
surface current that actually influences the deep sea. So it's not as deep
There
as
is no
here, but
waypoint
it's
3.
around 900 meters. We're seeing temperature changes whenever that Gulf Stream meanders over into
Yeah.
the region of where our temperature loggers are. So It just depends kind of on the local currents and how strong those are.
Well, and so especially where we're at, like right on the equator, one of the things that's an issue is that the temperatures are so consistent year round. Like even the surface temperatures are super consistent. So you don't get a lot of surface temperature change. So you don't get a lot of the mixing and upwelling that would normally bring like more productive deep water plankton to the surface.
Yeah.
And so that current hitting the seamounts and coming up, that upwelling we were talking about becomes really important in terms of more surface level life.
Right. Yeah, absolutely.
What's that guy we're seeing there on the left of the screen?
It looks like a dead sponge that has a bunch of brittle stars on it.
Oh, yeah, but sometimes works, sometimes doesn't. That's right. Have you noticed that there's, we're not spotting the little corals every now and then?
Yeah. Kind of transitioned over that hump and we aren't seeing quite as many as before. There's a small sea star on the right, if you guys see that.
Yeah.
Little cookie star.
We've moved on to the rough neighborhood.
Only the sea stars survive.
I like to think of it as rural.
We're out in the country.
Yeah. This appeals to my country heart.
Nice hiking trails.
Yeah.
Dirt roads.
Apple picking.
So what's the substitute for a deep sea apple? Like what?
I was just going through that in my head.
I got nothing.
Maybe it should be berry picking.
But I
What's
mean,
the
what are the
same
berry?
Well, I feel like there's more like
More likely to be a bush
small
than an
little
orchard.
like bushels than like large
Okay.
fruits.
Yeah. Oh, we're back to the trees.
It's a really tree-themed group.
Yeah, what's happening?
I mean, did somebody just read Overstory or something?
Very crunchy watch.
I just love berries. What can I say?
Yeah.
I have a student from back home who commented in that they broke one of my graduated cylinders in the lab, so
Oh no.
decided to hand
no.
out some detention.
I like how he tells you. I like how he tells you
Right.
that through the Nautilus
Yeah,
chat.
it's a safe
I feel
distance.
like that is
Yeah,
that's a
right.
safe place to make that.
He's like,
that
Brandon's
confession.
having a great time right now.
This is
I'm
a
gonna
good
tell him
time
that.
for me to say it.
Brandon, speaking of your students, I love your job and what you do. I think you should share that with our audience because you have like a really cool career.
Well, what I have is a terrible concept of work-life balance, really. So I teach part-time in the morning. I teach physics and chemistry, and then the second half of my day is I work at NASA's Jet Propulsion Laboratory in the education department, which is a really cool gig. I feel as someone who gets to see amazing robots being built that go up to space, it is incredibly cool to see robots that go to the ocean floor.
Yeah, and there's some collaboration now happening between people that build ocean robots and space robots, so that we can explore space oceans.
Space ocean.
It's a perfect union.
I mean, one of the
Ocean worlds.
Yeah.
The cool things about being on board the Nautilus is like all the different incredible science backgrounds. So I think in the mess earlier this week, I was having a conversation with one of the biologist who does diving work underneath the sea ice in Antarctica and all the ways that environment is actually similar to other planets. And so there's so much that we can learn when we're taking a look at these really extreme environments and bringing together all these disciplines to kind of share perspectives and share research.
Yeah. Yeah, we get a really unique opportunity to essentially explore another world within our own world when we get to do this type of work.
The condition we were saying earlier, the conditions for life down here are so dramatically and fundamentally different from what we expect and what we experience in our terrestrial lives that it is another planet.
Yeah.
Yeah, it also shows just how close-minded we've been as humans in terms of what life would look like if it were found somewhere else.
great.
The idea that it's going to be
Like
something
us.
that looks like
Yeah.
us, right? It's sort of a very egocentric argument.
One planet.
Yeah, we have definitely dropped off in some of the abundance of corals, which is interesting.
Thank you.
Why do you think that is?
It could be just a shift in, or like a change in orientation with the current. So we
Yeah,
kind of came
okay.
up a hump,
Yes,
and then
that makes sense.
the current's just flowing differently across this surface of rock.
Right. That'd be so cool to see, like, you know those models that NASA has of like surface currents from satellite data? They're really beautiful, or even just like weather models, like windy. To see that in like 3D of the current down here,
It would be amazing.
because it would be so different. You know,
Oh,
like
yeah.
even in like the little pockets of the rock,
Yeah.
like, you would notice so many different things.
Brandon, can you get NASA working on that?
Get
for us,
on,
please,
get on
please?
that one.
An ADCP on every mount.
Yeah. In every crevice.
Every crevice.
and coal map everything, like 3D map everything. And
Well, that's a given.
That's a really
That's a
good,
given, yeah.
but that brings up a good point too, Megan, is that like we are used to seeing some really great models from NASA, from satellites,
Right.
but all of that is surface
Yeah.
information because the satellites can't penetrate water to any significant depth. So it's actually really difficult for us to acquire all that information from across the water column. And it requires a lot more like manual sampling. So when we come out with other vessels, we'll do full CTD casts, which are devices to collect water, and that you can determine the temperature, the conductivity, depth, salinity, and stuff like that. But that's also just one profile of that spot. You don't
Right.
get that information from the whole place.
Yeah, it would be cool to like, get like specific profiles around something like a seamount, and then map it so
Could
so
we
well.
take a look at the little coral that's right there?
Little
The
coral.
little
A
one?
little one,
Surprise, surprise.
only 10 centimeters.
You want to zoom in?
Yep, let's go ahead and zoom in.
And is that a little shrimpy next to it?
It is
Yeah.
a little shrimp.
Hi.
Oh,
Little
All right, it's all
crino.
you, Marley.
a baby crinoid.
Is that a scale marm on that?
There.
Oh, yeah.
This is what I'm talking about with the sense of scale.
Yeah. Yeah, because that's like 10 centimeters.
Yeah, it's a little one.
Not large.
This looks like a plexaurid to me. So earlier they
That's a
sampled
good question. Is there a chance we're going to want to sample this?
yes, if you give me just a
Yeah,
second.
I'll hold off.
Okay. We might even need to step back.
Then. Okay.
Yeah, so I think if we're able to, this would be a good candidate for a sample.
Copy.
And Steve
What was
says
your last
the
step?
coral we saw on the left is a Rodana ritogorgia.
I would
so that's
do,
the one
yeah, I
that
do
has
at
a bit
least
more of a
20 meters
wavy
back,
axis.
probably 40.
Steve
'cause-
Oskovich is
right.
in our chat right now.
So
he's
ours
at
is gonna-
Temple University.
Yeah, but
we are
this is
lab
20 meters
mates,
grid
but
line.
he's really the
Right, so I would come back.
king of Pacific coral IDs. So it's great to have him on the line with
Is
us.
that an official designation? Does it come with like a special hat
It
or a
might be
scepter?
just in my world, but I think he's pretty up there as far as people that are able to ID these things specifically.
Okay.
Yeah.
I think
Should I make him a crown?
think
and
All right,
you
video, let's
should.
come full
Yeah.
wide.
Come in full wide.
Don't tell him that. He's listening, but don't
yeah,
tell
no,
him.
he is. Yeah.
Correct.
You're welcome, Steve.
Well, the layback would be just from here to here, right?
We're full wide,
I
Greg.
love that
Thank
still
you.
life
Yeah.
right there on the screen with the shrimp.
There's a little scale
Oh my
worm right
gosh.
there.
Uh-huh.
That would be a beautiful painting.
This is beautiful. Can we snap that?
We've snapped it.
I think
It's done.
somebody.
It's been
yeah.
snapped.
It's been snapped. Okay.
It's been
Scram
We're
it.
snapping continuously.
Continuous snaps.
Steve says he has a crown. It's in the data lab drawer. Sam knows what he's talking about. I don't know what
But
that
I
means.
don't.
Oh, I know what it is. I've seen this crown before on Steve.
He says, but I don't like titles.
Well, that's the sign of a true monarch.
Yes.
No labels.
No
Don't label me.
labels. The Argus view is really cool right now. You can see that large
Oh,
Rodana in front of Hercules.
wow.
Yeah.
That's a
let you
that's
know
a
when we need to move.
large.
It is. It's a
Cool.
big.
Yeah, we have a ship move coming back towards you, but
Yeah.
I'm going to watch it.
Got a Herc for
Yeah.
snail.
It looks like I can kind of wrap around if need be real quick.
Alexis, some people are wondering online
Yeah.
about the
That's actually going to be a little
the
far.
brittles kind of captured there on the coral sample. Can you comment on
Sorry, say that again.
the
Brittle star?
captured there on the coral? And
Yeah, so it's not captured by the coral. They actually use the coral as a way to get up into the water column and potentially filter feed themselves. So we see this pretty often where you have these brittle stars or ophoroids wrapped around corals, and they don't seem to be feeding on the coral. They don't really harm the coral in any way, but they just use it as habitat.
Okay, science. So there's no real way to just get just one branch of this. So I'm assuming we're going for the whole thing.
Yes, that works.
And do you have preferences to where it goes?
Just not with the Acanthogorgia.
Yeah. So we can put it in the forward box. We do have a sample in each of those, but they're totally different.
Okay.
And then for the starboard box, all 4 of the little ones are free.
I feel like that shrimp is just watching.
Oh, man.
What's going on?
We mounted the jaws upside down.
man?
What's that?
We mounted the jaws upside down. Right? So if I do continuous wrist rotate, that's as far over as I can go. And if I go the other way, right, now the jaws are upside down, but that's my center.
Oh.
Right? So I'm stuck here now. How you doing on?
I'm all right for now.
All right, video, let's push in.
Pushing in.
Greg, where are you thinking of putting this thing?
Forward box.
Copy.
Hold that.
Wow.
Got you in bubble.
Thanks.
The ship move's done. So
Better.
nice.
All right, full wide video.
Coming full wide.
Rack back.
We know we want the bin.
All right, we're full wide.
Okay, you can go ahead and bring out the bin.
Better coming out.
Greg, you can turn the porch light on if you want.
Ooh. Are there floaty bits in any of these boxes?
There's the acorn worm. I think
There's
is in
an
the other
acorn
one.
worm
In the
in
left.
port,
Yeah, it's right here.
and is it still there?
Yeah.
And just that rock in starboard.
Nicely done.
Yeah, really nice.
Going in.
Yep.
Nav, that's number 7 if you want to drop a
Okay, great. Thank you.
waypoint.
I would make a note to change that between,
Yeah, I'll write it here.
So is that a matter of how the jaws were mounted?
yeah, yeah. Okay, so let's turn. Yeah, so I rarely use the continuous wrist unless I'm actually just trying to spin around a bunch. And everybody's different, but I find that's much more natural for me.
That's fair. 04040, roger.
Okay, cool.
Yeah, I like actually using the continuous because then, like, especially when we're doing the camera shots and whatnot, obviously for that, but
Yeah.
yeah, everyone's different. That's
Yeah.
really pretty with all the contrast right now.
Yeah,
Well,
right.
we have the tether. Video, have fun.
Can we turn the lasers off for just a second?
Yeah, you got it.
Sure.
Yeah, I just put in a ship move.
So you got some time.
Yeah.
That's really pretty.
Ooh, that's
Kind of
nice.
reminds me of a willow tree.
It does.
And we're back to trees.
Wow.
This is very relaxing to
I
watch.
feel like I'm a bird.
Yes.
You're a fish.
I'm a fish, sorry. We're just land people, but we can't
No,
help
stop it.
No, sorry, we're fish. Channel fish.
Six
This
hours
talk's really
later,
nice,
and we're
I know.
all just still watching.
Mesmerized.
I love seeing the axis
Yeah.
and how it's like all those small.
Speckled.
Yeah, I don't know what
man.
Yeah,
What are
the
those little
detail on
speckles?
the axis is pretty really neat.
It's like furry.
Oh, now video's
Oh
showing
my
up.
God.
Yeah.
Oh.
Video is
Oh,
still
wow.
learning what she's doing.
Oh, she looks learned to me.
Oh man.
Look
So
at those
cool.
speckles.
Yeah.
Oh man. It looks like a tongue.
Yeah, it
Looks
does.
like- right?
It's
it looks
too
like
perfect. Oh my gosh. It's so beautiful.
I like how this coral was like trying to get in our view the whole time too.
it was. It was just like, will you please look at me?
Look
Yeah,
at
how are you going to
I don't know
sample that
what
guy
you're
next to me
looking
and
at.
not me?
I know you're gonna take that tiny little 10 centimeter plexar.
I've been here for thousands of years waiting for this moment. Give it to
Maybe
me,
hundreds.
guys. Maybe hundreds.
Make
yeah.
me famous.
Everybody
I'm
wants
old.
their 15 minutes of fame.
Wow.
We can at least give him 5.
Yeah, well, at least give them 5.
I don't think so.
This coral will live on forever.
You're famous, buddy.
We will tell your story.
All right, that was fun.
Yeah, we're good.
Thanks, Greg.
Great.
All right.
Thanks, guys.
Time to move.
That's the Iridogorgia?
I think it's Rodana-Ritogorgia.
Rodana-Iridogorgia.
So science, it is going to be very difficult to get a rock here. So
Yeah.
I think that when either Jess or I spot one that is actually grabbable, that seems like it's actually not manganese crust, we'll just let you know.
Sounds great. Thank you.
Do those spirals always spiral the same way?
All right, let me work my
That's
way
a
back
little-
over. We'll
I
get
don't
you on the
know.
right
I
heading.
think so. I mean, I think it's.
Like, are there lefties and righties? Is it
Yeah, I
the-
don't know.
Yeah.
When you're in the northern hemisphere, they go one way. When you're not in the southern hemisphere, they
That
go
would
the
be
other.
amazing if that was true.
Oh.
Coriolis effect in full effect.
Yeah, I never really thought about that for collecting rocks out here, but if you go into the field with a geologist, like they always have a hammer because you got to get down into the sample to get away from the crust, and there's no way for us to do that here.
No.
Yeah, that's one of the like big challenges that people are working on is like ways to develop drills that can do that underwater. But it's just everything is a little more complicated when
Yeah.
you're
I have
a
a,
couple thousand meters.
I guess, a redneck alternative to the high-powered drill. But
The crowbar of science?
no, I want to get a flange spreader from the oil fields.
Oh yeah, yeah.
You
Do you want to
know,
explain what that is?
Sure. So it's basically a wedge that as you, and it can be hydraulically driven, but we would probably just get one that was driven by a wrench, and we'd put a T-handle on the end, so then you'd use a continuous wrist rotate on the arm, and it basically goes from about 1/4 of an inch, 5 to 6 millimeters, to 30 millimeters with about 9 tons of spreading force.
Wow.
You can get heavier, dutier ones that do even more spreading force, but I figure, you know, just grab that, stick it in a wedge, like in a crack, and twist and see if you can break off a piece of rock.
Huh. Oh, that's cool.
Yeah, it's an elegant solution.
Wow, it's an inelegant solution to
I don't know. It's simple.
Yeah.
Hey, science, this is Invisible Ed up, not on video. Hey, Peyton, one of our ocean science interns that was just out with us recently in Samoa, was asking about that organism we were just imaging, if it was a Chrysogordiae? Coral?
Chrysogorgia. Yeah,
Yeah,
Chrysogorgia.
Gorgidae.
Yeah, so yeah, it's Rhodaniridogorgia, which is within Chrysogorgidae.
Okay,
So,
great.
yeah, good ID.
Nice eye, Peyton.
It's like people come out here and learn something.
That's why I still keep coming back. I refuse to learn. We've only had one screen rest, you know, like the eye rest where it just goes black for a little bit.
Yeah, that's a good point. It's like when it's feeling sensitive and temperamental.
Nice.
Well, it's had a lot of time to rest over the past week. Maybe it's just, you know,
So
it's like I'm refreshed, I'm ready to go.
Yeah, so it looks like we're coming up on this like mini summit, 20 meters, 30 meters
'Kay.
high summit.
Yeah, and
Okay.
Jess has mentioned if you spot a rock that looks grabbable,
Right.
it looks like it's basalt,
yeah.
shout out. The added bribe is that you get to grab it.
I'll look for any rock at that point. Look, there's a
It's just
rock
like
right
it's just like that one's perfect.
that looks, maybe it's fused, but I can try for that one.
The laser's back on. Try to give some perspective.
Yeah.
Actually, that cave might have been
Is my
like
headset off?
an
No.
ideal spot.
Okay.
Actually, yeah. 'cause
Just
there's more than
checking.
enough fragments in there.
Right, and there's just a chance that it's not as crusted. All right, sampled and back on our way.
Oh, is that the rock pen there in the lower left?
Maybe.
Oh.
Nice.
Good eye. That is a rock pen. So let's do some nice imagery of that.
Okay.
and then that would be a candidate for a collection if we can.
Okay. All right.
Ben,
So
you wanna back the
Yeah,
ship up?
1 second.
All right, so we're zooming in.
stand by.
Looks like it's scratching its back or something.
Well, there's nobody around it to scratch it for it.
Right.
Okay, video.
All right, going in.
Have you ever sampled one of these before?
A rock pen? No, I've never seen a rock pen before.
So this is a,
Scrape?
since we have the slurp on it, I would do a slurp and scrape
Go. Yeah, let's do it.
to use the technical terms.
It's really descriptive.
That's a really pretty.
The trick though is that Jess is really interested in the base, and the base is the hardest part to get.
Yeah. See if we can scare them off.
So Alexis, this is what we were talking about earlier with the rock pen, that normally they would anchor in the sediment, and this one is instead sort of suctioning up onto the rock, and so that's what we want to grab that guy.
Yeah, yeah, so
I want to suction it to my hand.
the other C pen, it's also a C pen, it's just, they're called rock pens because they
Greg, should I go
attach
ahead
to
and pull out?
rocks.
Yeah, let's come full wide.
And yeah, I agree with you guys that this section is probably the best, our best attempt to get the whole thing.
Some people online are wondering if we're
Do you mind that?
all
Yeah.
here on
Yeah,
the
and
UV
if you want
Nautilus,
this chair,
or if
you can
we're,
use it.
if we're remote. We're about 10 of us
Oh.
here in the
Okay,
research
stand-
van.
Stand by. Steve said don't suction it. They tried it and it was surprisingly rigid.
Okay.
This one.
So they want to go for a
For a grab?
grab. Let's see.
Ben, do you want to
Back up.
do a little bit back?
Yeah. Okay.
Like,
I do 10 meters.
220, 10 meters. Okay.
or 20? I mean, honestly, it won't really hurt.
Yeah, it's true. We have a massive.
So, what's the verdict here?
Well, I was gonna say, hey, why don't we switch and I'll just pilot Argus so that way you don't have to worry about it while you're.
Roger, thank you. All right, Alexis. So what's the verdict for us here?
So I think the goal is to grab it instead of using the slurp.
Grab?
Now, what you may want to try and do is nudge the base a little bit with one of the claws.
Try a Bob Waters
before-
move.
and just see if you can get it to, if you can agitate it a bit.
Yeah.
Are these guys like buoyant? If we knock it too hard, is it going to float away?
They're like just slightly heavy, so they'll kind of like come
Alright,
up a
video,
little
go ahead
bit
and
and then
push in
just
there,
kind of
please,
like
a bit.
drift their way back down.
Video, go ahead and push in a bit.
Yep, going in.
That's great right there. Thank you.
And I've got you on bubble as well.
Roger.
Nice. He's loose.
He is loose and floating now.
There's the buoyancy question answered.
Yeah, thanks, Greg. Come on.
Sorry.
You good?
And overdone again.
All right, now that's
I can't go over anymore.
rudder.
Yeah, you're out of.
Do y'all want me to zoom out or anything?
Yeah, you want to come wide, please? Probably behind us now. You can do a fast wide if you want.
So he is in us.
Underneath us now.
Okay, so he's just went around the corner. So
Oh,
let's
yeah.
pick up and back up.
Yeah.
Oh, nice.
All right.
Oh. Current is pushing him right into you.
Yeah.
Look up based on your.
Yeah.
All right, so I think next time we will do a slurp and scrape.
Yeah.
Because that came off pretty nicely.
Yeah, that was pretty good.
The ship moves over.
Roger.
What?
The ship moves done.
Roger.
Science, I don't know if you copied. Next time we are going to do slurp and scrape.
Yeah, I hear you.
And he will find a new home, or she. Oh, you're still hunting.
I'm still gonna get this one, I think. Just let it settle out for a
Yeah, it
second.
looks like it's.
Just slurp it.
Okay, I'm going to come around.
Let's go right about. Safely stabilize.
Yeah, we're in an ideal position with Argus, so.
Nice. Kind of on a marble here, but should work.
I gave you a bubble on the big screen.
Roger, thank you.
All right, so let's clear your slurp and then rotate to the right jar.
Roger, you can